Mitosis is a process of asexual reproduction in which the cell divides in two, producing identical copies with an equal number of chromosomes in each resulting diploid cell. Meiosis is a type of cellular reproduction in which the number of chromosomes are reduced by half through the separation of homologous chromosomes, producing two haploid cells. Mitosis is used in cellular reproduction throughout the body, whereas meiosis is only sexual reproduction. The cells produced in meiosis are genetically different, whereas the mitosis cells are identical. In meiosis, there are two cell divisions, and only one in mitosis. In mitosis, there is no pairing of homologous chromosomes, but in meiosis, there is. In mitosis, two diploid daughter cells are produced, and in meiosis, four haploid daughter cells are produced. The chromosome number remains the same in mitosis, and the number is reduced by half in meiosis.
Mitosis and meiosis are both very important to living organisms. Mitosis is responsible for DNA repair when cells are damaged and cell growth in general. Meiosis is pertinent to sexual reproduction. Here is an overall picture of the cell cycle. The cell cycle, or cell division cycle, is a series of events that takes place in a cell, leading its division and duplication. The cell cycle can be divided in three periods: interphase, during which the cell grows, accumulating nutrients needed for mitosis and duplicating its DNA, and the mitotic phase, during which the cell splits itself into two distinct cells, often called daughter cells, and the final phase, cytokinesis, where the new cell is completely divided. The period between mitotic divisions, that is G1, S, and G2, is known as interphase. In G1 phase, metabolic changes repair the cell for division. At a certain point, the restriction point, the cell is committed for division and moves into the S phase. In the S phase, DNA synthesis replicates the genetic material. Each chromosome now consists of two sister chromatids. In G2, metabolic changes assemble the cytoplasmic materials necessary for mitosis and cytokinesis. Finally, in the M phase, a nuclear division is followed by a cell division, cytokinesis. In G2 of interphase, a nuclear envelope encloses the nucleus. The nucleus contains one or more nuclei. Two centrosomes have formed by duplication of a single centrosome. Chromosomes duplicated during S phase cannot be seen individually because they have not yet condensed. In prophase, chromatids in the nucleus begin to condense and become visible in the light microscope as chromosomes. The nucleolus disappears. Centrioles begin moving to opposite ends of the cell, and fibers extend from the centromeres. Some fibers cross the cell to form the mitotic spindle.
The nuclear membrane has now dissolved, marking the beginning of prometaphase. Proteins attach to the centromeres, creating the kinetochores. Microtubules attach to the kinetochores. The chromosomes begin moving.
In metaphase, spindle fibers align the chromosomes along the middle of the cell nucleus. This line is referred to as the metaphase plate. This organization helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome.
In anaphase, the paired chromosomes separate at the kinetochores and move to opposite sides of the cell. Motion results from a combination of kinetochore movement along the spindle microtubules and through the physical interaction of polar microtubules.
Telophase, two daughter nuclei form in the cell. The nuclear envelope reappears along with the nucleoli. The chromosomes have become less condensed. Mitosis is now complete. In cytokinesis, the cleavage furrow forms, which pinches the cell into two.
The first phase of meiosis is called prophase 1. The chromosomes begin to condense and the homologs loosely pair along their lengths. Synapsis occurs and a tetrad is formed. Each tetrad is composed of 4 chromatids. The chromosomes thicken and detach from the nuclear envelope. The centrioles migrate away from one another and both the nuclear envelope and the nucleoli break down. During this phase, crossing over may occur.
The next phase is metaphase 1, where the pairs of homologous chromosomes are now aligned at the metaphase plate, with one chromosome in each pair facing each pole. Both chromatids of one homologous chromosome are attached to kinetochore microtubules from one pole. Those of the other homologous chromosomes are attached to the microtubules from the opposite pole.
In anaphase 1, the breakdown of proteins responsible for sister chromatid cohesion along chromatid arms allows homologs to separate. Unlike in mitosis, sister chromatids remain together after homologous chromosomes move to opposite poles. In the next stage, the spindle fibers continue to move the homologous chromosomes to the poles. Once movement is complete, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of 2 sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. Cytokinesis occurs at the same time as telophase 1, forming the 2 haploid daughter cells. The cleavage furrow forms. The genetic material does not replicate again once this is complete. For meiosis 2, meiosis 1 essentially repeats itself. During another round of cell division, the sister chromatids finally separate. 4 haploid daughter cells result, containing unduplicated chromosomes.